sir. Yes, sir.
Okay. Yeah. So yesterday we were discussing about the classification, or why should we have classification, or why should we have a subject or a branch like taxonomy, or a classification of insect, or what is all the uses of this classification. So we know that there are three important things: taxonomy, systematics, and classification. So we discussed a lot about all of that. So we discussed about so many things. Okay. So we said classification, it is nothing but grouping; taxonomy, giving names according to a law. Okay. So it is just we are applying a rules and procedures. That's all for grouping the organisms. On what basis we are doing systematics, where we are bringing in lot of relationship among them. So we are looking at the ancestors. So what is happening? What were the forefathers, or what is the today's species, and what was the species some million years ago or some 100 years ago? So we are looking at some kind of information. Systematics is the scientific study of kinds and diversity of organisms in relationship among themselves. Okay. So systematics, please don't get confused. They might ask you for the definition, define systematics, define taxonomy. So you should be able to tell them very, very clearly. Taxonomy is nothing but it is a theoretical study. Systematics, it is a scientific study of kinds and diversity of organisms along with their relationship. Whereas in case of taxonomy, you're only looking at theoretical part of grouping them into different species or organism or a family or order based on the principles, procedures, and rules. So both of them are two faces of a same coin. Okay. So why do you want systematics? So know about living organism, provide means of communication. It is a storage. It is a means of storage of information. It is developing lot of other things. So if you want to conserve, we should know the name. We should want to know their ecology, behavior for all these studies, or even you take toxicology. So any kind of entomology related thing, you need to know the name or the systematics. So why do you want to collect insects? So I think you all have been given insect collection, so you know how to collect insects. So I think I dealt all those things or explained why there is a need for study of insect systematics. Okay. So many of the insects which come from countries are becoming a serious problem to our own self. So that is also an issue. Okay. So we should be aware of what these organisms are, why from where they are coming. So to take up the control measures. So as a farming community, as an agriculture entomology, or as an agriculture students, so you all should know all these things which are very, very important when you take up the jobs or when you take up different responsibilities as an agriculture graduate. Okay. So that becomes, anyway, it is systematic. Okay. So all those things. So diversity, there are five insect orders which are quite diverse. One is Coleoptera, Lepidoptera, Diptera, Hymenoptera, and Hemiptera. So the 5 major insect orders. Okay. So they are very, very important. And okay. So coming to levels of taxonomy, there are 3 levels. One is alpha, beta, and gamma taxonomy. Alpha taxonomy is concerned with naming of the species. So yesterday we discussed that. So how do you name a species? So you have certain sets of rules. Okay. So you are going to name the species. Coming to beta taxonomy, it is concerned with the higher classification. So what you do is that you classify them into different types, orders, or suborders, or family level. Okay. So then it becomes higher level classification. Gamma taxonomy, it is concerned with the evolutionary relationship and phylogeny. And you are looking at very, very close relationship among the populations. For example, there are subspecies or varieties or some population variations. Okay. So you are looking at even the minute details. Okay. Then we were dealing with nomenclature. Nomenclature is naming of the species, where what is the difference between a common name and what is the difference between a scientific name. So how do you write a scientific name? You all know how to write a scientific name. So it is the genus and species. Genus name should always be in capital, and species name, which is again both genus name and species name, both should be italicized or written in. If you are writing, you need to underline them if you do not italicize. So what is the use of scientific names? It gives uniqueness, distinctive, and universally accepted, and it is the one of the stable ways of using the names. Okay, their names don't change, whereas the common names, they keep on changing according to the region, according to the language, or according to the different countries. Okay, so it all varies. So that's why we have a scientific name, so that we follow it on the entire world. It follows the same name. Okay, so what is binomial nomenclature? Who gave it? So it was Carolus Linnaeus in 1758. So where he published the this 10th volume of Systema Naturae, which is very, very important for naming of species. So that is very, very important, and his contributions are very important in the classification. Okay, so scientific names. So why it is, what is the difference between a common name? So yesterday I explained in detail. Okay, so any doubts on yesterday's class? Okay, we were telling about holotypes and paratypes. We also discussed about holotypes and paratypes. Holotype is a single specimen. Paratype is the species, other specimens which are collected along with the holotypes. And holotype is always, it can be a male or female, and paratype also can be a male or female. And holotype is the first specimen which we are using it for describing it as a new species. Okay, and it is given a red label, whereas paratypes are given with a yellow label. So have you taken any seed technology course?
No, sir.
Okay. Anyway, so again, they use different color on the seed bags. I think most of you should be knowing that. Okay. Anyway, I'll not confuse it. Okay. Holotypes, paratypes. Okay. So that is very, very important for your courses. As I said, holotype is the single specimen which we put it in a red label. Okay. And the paratype in a yellow color label. So that is what is very, very important. And you know, this person, Aristotle, so his contribution, so he is one of the first person to start or describe species. Then comes the contributions made by Linnaeus. Linnaeus Systema Naturae, very, very important, one of the milestones in the entire world, where Systema Naturae 1758 10th volume was published, where plants or animals are given names according to the set of rules and regulations that we follow. Okay. Then this is Linnaeus. Okay, so we discussed a lot about him. And binomial nomenclature, genus name, species name. So, and you know why we have to have a nomenclature: uniqueness, universality, and stability. So I think okay. Okay, so coming to species concept. So we were dealing with biological species concept. A species, you all know the definition of species, right? Anybody? Can anybody tell me what is the definition of a species? Can anybody tell what is the definition of a species? Species is a group of organisms capable of interbreeding among themselves. Okay, if you take any group of organisms, they should be able to interbreed among themselves. Then only we call them as a species. Okay, so that is the definition of species. Definitely they will ask you for 1 mark, define species. A species is a group of organisms which are capable of breeding or which are capable of interbreeding. Suppose if you take there are 2 populations, okay, and they mix up, but they cannot interbreed. They can be together, but they cannot interbreed. If they interbreed, then only they become a separate species. Otherwise, they are not a species, and they should reproduce, and then the offspring should be generated. So then only they become a group of organism or a group of organisms. We call them as species. Anyway, so coming to species, so we have species is one of the last line. Okay, if you take a species, we have how do you say that? Okay, if you take a species, so you can say that it is one of the last level, okay, of the species. Before species, what is there? Anybody know what is that before species? Before species, we have genus. Before genus, we have subgenus. Okay, and before subgenus, we have the subfamily. Okay, before above subfamily we have family, and above family we have subfamily, and subfamily, family. Okay, then above that supra. Okay, I'll put it like this. Okay, I'm sorry. Okay, you know the taxonomic hierarchy. So this is they will ask you to write the taxonomic hierarchy of an insect, for example. So what comes first? Kingdom, then subkingdom, then we have phylum, then we have subphylum, superclass, then we have class, then we have order, we have suborder, then we have superfamily, family, subfamily, then we have tribe, and then the genus. Genus again will have subspecies, and subspecies will have the subspecies. Or this is how the taxonomic hierarchy goes on. Please remember: kingdom, subkingdom, phylum, superclass, class, and order, suborder, superfamily, family, subfamily, tribe, genus, and species. I think till here it is all common. Okay, so kingdom, subkingdom, phylum, superclass, and class. So from here there is a change in case of your insects. Okay, especially in the hierarchy of the insects. So you have order. So there are in order we have suborders. Okay, again in suborders we have superfamilies, family, subfamily, tribe, genus, and species. So please you should be able to write it. At least this part is very, very important for you to write it in your exam. Write the taxonomic hierarchy of the animal kingdom or insecta, they'll ask you. So you are supposed to write this is how it goes. For example, Animalia, then we have the subkingdom, invertebrata. Invertebrata means they lack the backbone. Or okay, then we have arthropoda. Arthropoda means jointed legs. Phylum Arthropoda. Insects belong to the phylum Arthropoda. Superclass Hexapoda. Hexa means 6. Poda means leg. Then class Insecta. Okay. Order. Order can be anything. How many insect orders are there? Anybody?
8.
8. Okay. Somebody, anybody? Okay. A few more answers. There are 29 to 30 insect orders. It depends on the person whom we are referring. Okay. Anyway, has it not been told in your practical class in the beginning how many insect orders are there? Okay. Anyway, there are 29 to 30 insect orders. Okay. So there are a lot of changes happening, but anyway, we'll not get into the complexity. But as of now, there are 27 to 30 insect orders. Okay. It might be 27 or it might be 30 insect orders between that, because a lot of changes are happening. But at least you should remember there are quite a number of insect orders. So you should remember all the characters of all these insect orders. So that is what is taxonomy. Okay. So we have, for example, Hymenoptera. Anybody know what is Hymenoptera? They are bees and wasps. Okay. Bees, wasps, and all the ants, they belong to the order Hymenoptera. Okay. Then suborder, Apocrita, superfamily, Apoidea. Apoidea is the superfamily. Family will be Epidae, subfamily Epinae. Then we have the tribe Epini, genus Epis, and species Epis dorsata. So what is important is that, so when we are writing this, they will ask you, write the hierarchy, taxonomic hierarchy of the, and with an example. So you should remember this example. So you see that the endings of the above the family. So if it is only family, it will be IDAE, IDAE, superfamily INAE, INA, and if it is tribe, it will be INI, and if it is genus, it will be the APIS. Okay, so you take only the few words, and the last 3 words. INI means tribe, INA means superfamily, IDAE is family, idea, OIDAE is the superfamily. So that's how it goes. Okay. So superfamily, idea. How do I tell it? Okay. Please listen. So what is happening here is I should not confuse you. Okay. So we have the order Hymenoptera. So they will ask you, write the taxonomic hierarchy of an insect order along with an example. If they ask you, you should be able to write this example. Suppose if you want to give an example, if it is the super family, the ending will be idea. So forget about AP, it will be OIDAE. If it is super family, if it is family, it will be IDAE. Endings, any name you can take, the ending should be like this. If it is for super family, it will be idea, it will be IDE for family, IDAE, and INAE for subfamily, and tribe, it will be INI. Okay, so that is how it goes. That is how the naming or the hierarchy is written. Okay, clear?
Yes, sir.
Is it clear? Except this year, I want the answers from others. If somebody is not clear, let me know. So what is happening is that in case of the super family, you take out this. Can you all see this? So it will be idea. In case of family, it will be IDAE. In case of super family, it will be only these three words. And in case of tribe, it will be this. So this is how it looks. Okay. For example, okay, AP, we are taking the stem AP, apoidea, APOIDA. If it is family, it is APIDAE, epidae. If it is subfamily, APINAE, epinae. If it is tribe, APINI. Okay, so that is how this names all this 1, 2, 3, 4, and 5. So especially the family, super family, subfamily, and tribe, all these things will be having this common ending. So you can easily identify whether it is a family, it is a subfamily, or a tribe. Clear? And you know, the genus is always written in capital A with italicized, and the scientific name will have two, or the species name will have two names, that is Apis dorsata. Apis dorsata is your rock bee. Please remember, the rock bee is Apis dorsata. Clear now? Are you all there?
Yes, sir.
Okay. If you are there, is it clear?
Yes, sir.
Okay. Okay. So there are few definitions which you should know. Species, a group of interbreeding population. Genus, all species with similar character. Okay. A group of genera is called as tribe. Family is nothing but a group of tribes. So this is how we define the species, genus, tribe, and family. Okay. Then coming to the definition of a species. So if you see this photo, do you think it is a species? All of them are same species?
Sir, it's of termite on.
Yeah, termite only. But they look very, very different, right?
Sir, it's only the stages are different, but ultimately we have to recognize from the final stage only.
Yeah, so they are not stages. Okay, they are different morphs. Okay, so we see that it is a species is a reproductive unit, or it is a genetic unit, or an ecological unit. Okay, so therefore species is a group of interbreeding populations, so they can interbreed in that and are reproductively isolated from similar other groups. So then only it becomes a species. Yes, absolutely, I agree with the answer. So it is a queen termite. You can see the physogastry, the abdomen is very much enlarged. So it is the king, it is the soldier, and it is the worker. but all of them are a same species, though they look very, very different, but they are same species. Why do we say they are same species? Though they look very, very different, their mandibles, their abdomen, their wings, so lot of variation. But on one basis, we say that they are capable of interbreeding among themselves in the same population. So then only we say that it is a species. So this is the meaning of our understanding of the species. Okay. So there are few more definitions which they might ask you. I don't know how to tell you. Okay. Okay. So you know what is a species now? Yes, right? A group of
Yes, sir.
Okay, a group of interbreeding population. So what could be the subspecies? So there is some variation. Suppose in one population, there is a slight change in the color. Some look very, very dark and some look pale. So that means they are same species. If you see that, if you look for the characters, then they are same, but only there is a slight variation. Okay, we say Anglo-Indians, we can say Negroes because we looking at the color, right? So we say they are from South India or they are from America, looking at the skin color, right? Or some kind of changes. They are same. They are also human beings, right? Yes or no?
Yes, sir. Yes, sir.
Yeah. So that's how if you go to West Indies, you see the West Indian cricketer. If you take Chris Gayle, he's very tall. He has a different kind of hairs. The color is dark. Okay. On the same way, if you take an England cricketers, they are completely whitish, right? Their hairs are completely white or cream colored. So, but they're all same human beings. So that means they are just the races. Okay. So that is what the subspecies is. Species is, I have explained that, reproductive community. So in case of the species, I was also mentioning about a reproductive community. The individuals of the species are able to recognize each other as a potential mate. Okay. Then we call them as a reproductive community. And an ecological unit. Irrespective of the number of individuals, they act as an unit and interact with the individuals of the other species with which they share the habitat. Okay. Reproductive community means they should be able to recognize their own same species for mate. Okay. Or they are regarded as the potential mate. Ecological unit means there may be so many species in individuals, and there might be also other species in the same unit. So that becomes a ecological unit. Clear? Subspecies, species, then what is a reproductive community and what is an ecological unit? So there are 4 definitions which they might ask you. If they ask, please, you should be able to write 1 mark. You can easily get with these definitions. 1 or 2 marks, you can easily get it. Okay. Okay. So that is where I'll stop at the taxonomy. But before that, I have few more things to show you. For example, I was telling you the naming of the species. Okay. All of you are there. So if you look at this, there is a slide which we see that. Okay. So naming of species means we give names for insects. So for example, you know who is she that? Can you all know who is this lady? Okay. I said that taxonomy is the taxonomy is naming of species according to certain rules and regulations. So you can name it is a mosquito, I'm sorry, it is a wasp which has been named after the this lady Shakira. So you can see that AE, it is a female. If the female is there, they will be adding AE. For example, if you want to name any species by the India, so what we do is that India is a mother country or a father country?
Sir, mother country.
Okay. Can you give an example for father country?
Don't know, sir.
Okay, find it out. Okay. So anything which we want to name it after India, we will name it as Indicus. Okay. Indicus. Okay. For example, Indicus means India. US is for the feminine gender. Okay. US and AE will be lastly added for female. Okay. If it is a male, it will be last ending will be I. Okay. It will be ending with last ending will be I. Okay. For example, what would be the name of this? You know this person?
Hitler, sir.
Okay. So his name will be ending with Hitleri. I. Last Hitler, the end will be after. In case of male, it will be ending with I. Okay. There are a few examples so that you should. Okay. All of you should be knowing this lady, especially boys. Don't worry. Need not be shy. Please let me know the answer. So who is she?
Lady Gaga.
No, she's not Lady Gaga. Wrong, I say. What is this? So it is Angelina Jolie. So you can see that the name ending with AE for if it is a female. Okay. So our name is Angelina Jolie. So then we have the ending, and she's been named after a spider. Okay. Anybody?
Bill Gates.
Very good. What would be his name ending?
Sir, Bill Gates, sir.
I know it is Bill Gates. What will be the ending of his name?
Gates.
Gates or Bill Gates? So it is Gatesy. So they have taken Bill Gates and they have added I at the names. Gatesy, Bill Gates is Gatesy. Okay. So Aristotelis is the genus name and the species name is Gatesy, named after the founder Bill Gates. Anyway. Okay. Now you should be able to tell this.
Donald Trump.
Okay. What would be the name, scientific name of that species? What will be the last species name of the moth? Okay, you can see that Trumpsy. Okay, very good. Banu has said that Trumpsy. Okay. Anyway, so what is happening is that this moth is looking, look at the top of this moth head. It looks like the Bill Gates or the Trump's head, and they have named it after him. Okay. So if you look at it, looking at that, so they have named after the president Donald Trump. Okay. Donald Trump. PI. Okay. So if you look at this, this is the moth which looks like Donald Trump. Okay. Somebody had answered. Okay. You can see that one more name, species named after Charlie Chaplin. Okay. If a fly is named after Charlie Chaplin. Okay. If it is a male, it will be. And you should be knowing this person. Okay. This is a well-known person. So David Attenborough, you all know him, right? Yes or no?
No,
No,
sir.
sir.
You don't know?
No, sir.
Oh my goodness. Okay. Who said no? I want you to meet me sometimes. Okay. I'll tell you who is. Okay. Or just Google, find some time, find it on the internet. Who is he? Okay. He is one of the very well-known persons. If you are all okay, many of the insects and many even dinosaurs, fossil dinosaurs, have been named after him because of his contribution to the earth. The contributions he has done to the earth is so much that he has done so much for the planet. Okay. So Sir David Sir is even a species is named after him. Sir David is based on his name. Okay. So anyway, these are some of the insects which I have named after my professors. Okay. For your example, you see that Viraktamatai. So Viraktamat is my professor. So I have named it after him. And there's one more professor. He is Dr. Belwadi. So I have named it after Dr. Belwadi because he is my professor. Or if you like him, if you like, you can name after your girlfriend, your boyfriend, or whatever it is. Okay. It need not be. Okay. And it all depends on, or if you don't like somebody also, you can still name them. Okay. After them. Okay. Anyway, so these are my professors. One is Dr. Balwadi, Dr. Viraktamat. Both of them are my professors. And one more person, Ramurthy, who was also a person who is a well-known entomologist. So I have named it after them so that they for their contributions. Okay. So anyway, these are very interesting insects. So which look like that. And if you look at this, there is also one more name, Pratapanai. I think somebody was okay. It is because again a professor from Kerala who helped me collect this insect, so I named it after him. Okay. So anyway, so there are so many funny things. Okay. So you can name insects the way you want, but only thing is that you should follow the scientific rules or the law. For example, somebody is named a fly after a pizza cake. Okay, piece of cake, pizza cake. So you can do it anyway, whatever the way you want. Okay. Anyway, I'll stop at this this thing. So we are getting into something else. So what I want to tell you is that, so I'll get back to my normal slides. So I have few things why entomological collections are important, why taxonomy is important. Please watch this video. Okay, carefully, please watch this video.
Okay.
Sorry, please watch this video.
Okay, I'm Lois.
And I'm Charlie. And we have been told and we believe that we have the largest private collection of insects in the world.
Yeah.
The story of how we met was interesting enough. I was a teaching assistant at the University of Arizona, and Lois was a student in the class that I was teaching. We've been married for almost 55 years.
He's got a million stories about insects, all of which I've heard and all of which I can fill in sentences.
And we've traveled extensively, have been to all the continents and 70 countries collecting. It fills us with pleasure to get into the field and collect, to spend the time in the field together.
Oh, you need to look at the antennae on one of these. It looks like a clown shoe.
Well, I study weevils, and the weevils are the largest family of living organisms.
These are plant hoppers, and I think their noses are cute. I collect plant hoppers, which are insects that feed on the juices of plants, the phloem or the xylem.
With piercing, sucking mouth parts.
With piercing, sucking mouth parts.
Oh, yeah, this is interesting.
When we get some insects in, they're just dead. Of course, we have to mount them, so we have to pin them or point them, and then we label them. Ah, the glue was not stiff enough, and he's fallen off his point. Then after I do that, then I sort them to genus.
Each of these units, trays, holds 55 specimens on average. And we have at least 1 million weevils and 250,000 plant hoppers.
Okay, here you come, Charlie.
We are donating the collection to Arizona State University.
And there will be research done on it, which is what we wanted to have happen.
When we were entomologists at the university, we worked 14 hours a day, 7 days a week. Now we're down to 8 to 10 hours a day, 7 days a week. It's a lot of work, but we both loved what we were doing, and it was sharing that was part of our life.
Yeah. Which one do you think is the prettiest?
These two.
I don't like that one.
Lois has often taken note of the fact that a very large number of entomologists are divorced.
What I recommend is if you're going to be a taxonomist, a systematist, collect insects, you get a spouse that is another entomologist.
Okay, if you look at their dedication, so they have been collecting a lot of these insects, and their collection is worth millions of rupees. Okay, and especially they become very, very important as a museum. So they are the reference material for the entire world. Okay, similarly, we have a small collection in our department. So if you think you can look at it, I would like to show you. Okay, is it fine?
Yes, sir.
Okay, so this is the collections at the Department of Entomology that I had prepared for a presentation. Okay, so in fact, it was a meeting where during the corona we could not directly talk to them. So what we did is that I prepared a video for the people who wanted to look at our collections. Okay, so anyway, So you are all aware of this, right?
Yes, sir.
You all know this building. Okay, this is the building which I was telling you, where the collections or the entomology museum is being kept there. Okay, so this is where we work or I work. Okay, this is for Namaskara. I welcome all of you to the virtual entomological collections network. So this is our lab. I'll take you around our lab and we'll get into our collections. So this is the facility that we have. Okay, so what we have is that collection facility where we have all these specimens deposited. This is our mobile rack system, wherein we have deposited all our collections. This is the facility that we have in our department. Okay, this is similar to the other museums, and it is one of the India's best collections we have. Okay. The collection is housed in the Department of Agricultural Entomology, University of Agriculture Sciences, Bangalore, and is supported by University with project grants from ICAR. I currently take care of the collections in an honorary capacity. At present, no full-time staff have been assigned to the management of the collections. So we do a lot of taxonomy, right? So we need microscopes. We have high resolution microscope where we take up images, where we make diagrams. So this is our where collections primarily include insects of agricultural importance, and we have about 270,000 specimens housed in this collection. Now it is quite around 3.5 lakh specimens. The oldest collection we have here is a weevil Archidodas collected by Leslie C. Coleman, a Canadian scientist who worked in the princely state of Mysore in early 1900s. We have about 700 holotypes and roughly 1,800 see the holotypes and paratypes collections by various researchers. So this was one of the recently described species of cicada. I have named a very large number of cicadal types are due to the work of Professor. This is my professor, an eminent leafhopper specialist. Is a world. During the course of more than 3 decades of his teaching, the collections made by graduate students have enriched the holdings. In addition, many of the students in the department have worked on various taxa under his guidance and have contributed to the collection. So this is our collection. Look at that. Ants, you take butterflies, you take moths, you take... One of the largest moths recently described as a new species from Western Ghats. Okay, so this is a moth which was recently described as a new species. Bugs, Atroptera, all these are bugs. The collections also include wet specimens, immature insects, and termites. So these are the immature There larvals. Are also orchard specimens deposited by researchers who work on insects associated with agro-ecosystems. Cicadas. Colemania spinoradis, an endemic species named after Leslie C. Coleman. Specimens of desert locusts collected during the recent outbreaks in India. Commonly occurring species of fruit-piercing moths in India. One of the largest stick insects from southern India. It is 21 centimeter. Large beetles, very, very large beetles. We have a separate section to house the mite collections. Collection facility helps in various ways, including teaching undergraduate and graduate students, training researchers, and for outreach programs to the general audience. We provide identification services to researchers, farmers, quarantine, and other government agencies. We were recently able to confirm the entry of African Phalaronum, Spodoptera fujiparda, into India in 2018. We also loan out specimens mainly within India. For researchers from other countries, the rules and regulations set out by National Biodiversity Authority need to be followed. Researchers can visit our facility in person. Not only that, we had a very big exhibition for general public. Look at all the insects displayed there. Thousands of kids, people visited that. We had live insects, we had dead insects. We have had numerous visitors to our collection over the years, and we welcome you all too. so there are lot of people who come from abroad. Okay, so this is a person from Australia. Okay, again a person from British Museum, London, a well-known leaf of bee specialist. Okay, so a lot of people come in. So they come and study, look at our specimens, and which help them in understanding. Mick Webb again is a well-known authority for the entire world. So people come here. So this is Dr. Belwadi, whom I was mentioning. Okay. Anyway, so this is what is our collection. If somebody is interested, please do come and visit the department. I'll be able to show you at least few collections so that you get interested in entomology. Okay. Yeah. Are you all there?
Yes, sir.
Yes,
Okay.
sir.
Okay. So did you all know that there was something like that in the department?
No, sir.
Okay. Anyway, if I would invite you all sometimes when you are free, when you don't have any classes, please let me know so that you can visit it. Okay. Anyway, so coming back to the classification or into the taxonomy. So now here afterwards, we'll be completely speaking about the taxonomy. So what we do is that, so now you have a background idea why we should have taxonomy or why we should collect insect and what is the use of collecting insects and pinning them, mounting them, keeping them. So it is not one day job, or it is for the future, or it is for the benefit of the entire nation. It is the national property. It is not just collecting them. So you think they are insect, we kill them or squash them and throw them. It is not that. Okay. Each insect has its value. Okay. So coming to with this background, all this background, I think now you understand why we need to look at insects very, very carefully. Okay. So now coming back to the course, the next practicals. So we have the practicals are the in future, there'll be insects which will be kept under microscope. Okay. So you will be looking them. Okay. How do you identify the insects? You have some character. For example, if you want to identify your CR, you have a certain character. He's pale, thin, very tall, or something like that, right? So you have a characters. You apply some characters and identify them. Okay. So that is why that is how you do that. Coming to the classification of insects. So we said that the class insecta is been grouped into is classified into two subclasses, that is Pterygota and Aterygota. Aterygota means without wings. Pterygota means with wings. Please remember the class insecta is being grouped into two subclasses, that is Aterygota and Pterygota. Aterygota without wings, Pterygota with wings. Okay. Please remember, you should be able to differentiate, right? The difference between a Aterygota and Pterygota, they will ask you. Aterygota means primarily wingless, evolved from wingless ancestors. Means primarily wingless and secondarily wingless. Anybody know what is secondarily wingless? They have wing pads, but the wings are reduced. Okay, so because of that, we call them as secondarily wingless. Or for example, your bed bugs. So they have wings. If you carefully collect them and see, they have wings, but they don't use it for flying, or they have been reduced. Because of that, we call them as secondarily wingless. Okay. Sudesh, you have any other class after this?
Yes, sir.
Which class?
Soil science, sir.
Okay. Okay. Anyway.
Sir, you can take 15 minutes, sir, if you want.
Okay. Yeah. That's why I need 10 minutes extra.
Okay, sir. No problem.
Yeah. Okay. So coming to Pterygota, we have primarily wingless. Means they don't have wings at all. Even you catch them, dissect them, you don't find any parts of the wings. So that means from the ancestors itself, they are without wingless. Whereas Pterygota, secondarily wingless, means there were wings, but over the period they didn't use them, or because of that they lost the wings, or they are not using that wings, or it has been reduced. Metamorphosis means growth, change in the growth. Whereas in Aterygota, there is no change in the growth, or no change in the, there is a change in the growth, but not the structure. Metamorphosis means not in the external look, they don't change. if for example silverfish, from nymph to adult it will be the same. Okay, so that means they look the same as the nymph and also the parents. Whereas in case of pterygota, the metamorphosis is present. In case of pterygota, the mandible articulation in head is monocondylic. That means a single point of attachment, whereas here in pterygota, there is 2 points of attachment of the mandible, so they can move. Then pleural sulcus in thorax is absent. In case of pterygota, the pleural sulcus is present. Then pregenital abdominal appendages present. Example, anybody, what are the pregenital appendages? Any example? Pregenital abdominal appendages. In case of columbola. Okay. Yeah. So those are the pregenital abdominal appendages. Yeah. So whereas in case of pterygota, they are not present. They are only present at the last. Okay. There are no appendages on the abdomen. Okay. So because of that, that is how you differentiate apterygota and pterygota. Clear? Class Insecta is grouped into two subclasses, that is apterygota and pterygota, based on the presence, absence of the wing, or so many other characters. 2 marks. Differentiate pterygota and apterygota with example. Okay, so you will get 2 marks easily. Or they might ask you metamorphosis is absent in apterygota or present. So you should not get confused. Okay, please remember, you should be able to write the answers for all these things. Okay, then. So characters, so wing development, for example, there are also other characters. Okay, coming to the pterygota, there are one more changes. Okay, again, somebody, okay. Oh my goodness. Okay, coming to the pterygota, the wing development is exopterygota. And okay, coming to pterygotans, we have 2 groups again, endopterygota and exopterygota. So in exopterygota, the wing development is external. In case of the endopterygotans, the wing development is internal. For example, complete metamorphosis. In case of this, it is hemimetabolous. In case of bugs, grasshoppers, the wings are developing outside. Whereas in case of the holometabola, for example, you have the butterfly pupa, complete metamorphosis is there. The wings are completely inside the pupa, right? You don't see them outside. And only when they molt and become adults, you get the adult the wings. So that means they are developing outside the body or inside the body. Exopterygota, for pterygota and apterygota and pterygota. In apterygota, we have 2 more classes, that is subclasses, that is the endopterygota and exopterygota. Exo means the wing development is outside the body, whereas endo means the wing development is internal. It is in case of exopterygotans, the incomplete metamorphosis is seen. Example, for your cockroach or your grasshoppers or any of those things, if you look at it, the wings are developing outside the body. Whereas in case of endopterygotans, the wing development is inside the body. Okay, they become pupa, and suddenly from the pupa you get a adult. Okay, the wings are developing internally. That means the wings are developing internally. Then coming to the pupal stage, it is absent in case of the exopterygotans. Whereas it is present because it is a holometabolous insect. Holo means all these stages are there: egg, larvae, pupae, and adult. Okay, so pupal present. Whereas in case of exopterygota, the pupal stage is absent. They are called as nymphs or naiads. Okay, so you can see that. Okay, the young ones are called as nymphs or naiads. Whereas the endopterygotans, they are called as the larvae. There are 16 insect orders in case of exopterygota, and in endopterygota there are 9. So nearly it is 27 insect orders, which we come at 27 to 26 plus 3, 29 insect orders. Okay, so there are 29 insect orders which we have in the entire insect. Okay, so this is what it is. Okay, there are few more groups of insects. Okay, so that means Paleopterans. Palea means primitive. Okay, whereas primitive insects. So there, the wing flexing mechanism is absent. Okay. For example, if you look at the dragonfly or a cockroach, all those things, there's some variation in their wings. Okay. There's no wing coupling mechanism or something like that in case of some of these insects. Whereas in case of paleopteran insects, the wings, they do not have the wing flexing mechanism. They cannot flex the wing. There is no muscle attachment which will be supporting the wing flexing. Okay. They are all both the wings are independent. Okay. Paleopteran, for example, mayflies, they are the primitive insects. Okay. So that they don't have this wing flexing mechanism. Paleo means primitive. That means they don't have the wing flexing. Orthopteroid group, okay, which look similar to grasshoppers. Grasshoppers, cockroaches, all of them, they have put under orthopteroid group, including their mouth parts. Okay, they have the hind wing well developed, abdomen is always with cerci, many number of Malpighian tubules and ganglia on the ventral nerve cord are fused. So because of all these things, we put them into orthopteroid orders. Then hemipteroid orders. So we have mouth parts, australate mouth parts, and sucking mouth parts. All of those insects, we put them under hemipteroid insects. Panarporoid orders, whereas in panarporoid orders, the holometabolous insects, and they have the larvae and pupa. Panarporoid means they have this kind of metamorphosis. Okay, so holometabolous insects are called as panarporoid insects. Okay. Anyway, coming to the superclass Exopoda, we have Exopoda belongs to the class
Insecta, sir.
Okay, very good. Class Insecta, superclass Exopoda. In Exopoda, we have two groups, that is non-insectan Exopoda and Insecta. Okay, so class Insecta, where we have non-insectan Exopoda. Why do you call them as non-insectan Exopoda? Is that because the antenna and mandible. So sometimes the antenna is absent in some of these organisms and the mandibles. Okay, so because of that, the mandibles are internal. Usually for most of the insects, the mandibles are outside the body, whereas for this non-insectan Exopoda, the mandibles are internal. Okay, so because of that, we call them as non-insectan Exopoda. Okay, what are those? For example, Columbola. You know Columbola, right? Colophore, retinaculum, the jumping one. Because of that, we call them as all of them, Columbola, Protura, and Diplura, we put them as non-insectan Exopoda. They are having six legs, but are 3 pairs of legs, but still they don't have all the characters of for example, Protura and Diplura. Okay, diplurans are the proturans do not have the antenna. Okay, because of that, but we still put them under insects. Okay, so all this, because of all this kind of modifications, we put them under a non-insectan Exopoda. They have 6 legs, but still we consider them as non-insects. Okay, but still they are insects only. Don't worry, because they have some modifications or some changes. Okay. Then coming to Columbola, what is that? There are only 6 abdominal segments. Yeah, otherwise there are 12 abdominal segments, 11 abdominal segments. But in case of Columbola, there are only 6. Because of all these things, we separate these 3 insect orders as non-insectan Exopoda. Okay, so clear? Okay, class and order Protura, class and order Diplura, class and order Columbola. Both class and order are same for this. Whereas for Insecta, the class is Insecta and orders keep on changing. Okay, so we have Ectognothus. As I said, the mouth parts are developed outside. Okay, we have Aterygota, means wings are absent. Pterygota, wings are present. And again, Pterygota has been grouped into based on the presence of wing flexing mechanism or the folding. We call them as Paleoptera, Neoptera, and then we have the Paleoptera means primitive orders, Epimeroptera and Odonata. Neoptera include all the other insect orders like Polyneoptera, Paraneoptera, and finally we have the something goes wrong. Oligoneuroptera. So this is how the classification or the grouping of the insect order goes on. Okay. Aterygota, Pterygota. So in Aterygota, we have Thysanura and Archaeognatha, whereas in case of these are the insect orders. Okay. Thysanura is nothing but your silverfish. Okay. Then in case of Pterygota, we have made them into 3, 4, and 5 groups or 5 divisions. Okay, there are 5 divisions in case of Pterygota based on the presence of the wing folding mechanism. Paleoptera means primitive; they cannot fold their wings or they do not have the wing flexing mechanism. Okay. Okay, coming to Neoptera. So what is happening with Neoptera? We group them based on the mandibles and wing development. So again, we have Polyneoptera, means poly means many, para means half, oliga, what do you mean? They are very, very specific. So you see that there is so much, all these are insect orders. They come in Polynia, Plecoptera, Blatodia, Isoptera, Mantodia. All of them, they come under Polyneoptera, many. Okay, so there are many. Para means few. Okay, so means half. So Oligoneoptera, they are completely, they have lot of modifications. I'll get into this later. So you can see that 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23. So 23 insect orders. So 23, 24, 25, 26, 27, and 3 non-insectan hexapoda. So they are nearly 30 insect orders. So you should be able to remember all the characters of the 30 insect orders. Okay. So I don't know how you are going to remember, but you need to remember them. So we'll see how you are going to remember it. Okay. So coming to the insecta, we have Pterygota and Aterygota. Okay. I said that. And again, coming to Pterygota, we have Exopterygota and Endopterygota. Exopterygota means hemimetabolous insects. Endopterygota means holometabolous insects. What is the difference between Aterygota and Pterygota? I said that this is the difference. Okay. I said that. Then coming to Exopoda, non-insectan hexapoda, there are 3 orders which fall under this. That is Protura, Dipleura, and Columbola. Okay. So what is that? So here afterwards, you'll be giving few characters which you should be able to remember. Okay, 5 more minutes. I'm sorry. Okay, so coming to Protura, class and order Protura, we call them as proturans. Okay, they are very, very minute insects. If you want to collect them, you have to collect them in the leaf litter. Very, very tiny fellows. Okay, minute insects with entognatous piercing mouth parts. Ento means internal. The mouth parts are all, for all these 3 in groups, the mouth parts are all internal. Because of that, we do not put them under the insectants. Okay, minute insects with entognatous mouth parts. And what you see is antenna and eyes are absent. You don't see 1, 2, and 3 pairs of legs. Antenna is not there. Okay, antenna and eyes are absent. Okay, and abdomen 11 segmented with terminal telson. So they have a terminal telson with a first 3 with a pair of small appendages called as styli, and 4 legs, sensory, and held above the head. So what is happening is that they have a terminal telson at the tip of the abdomen, whereas 4 leg is held like a sensory organ, just like the antenna, whereas eyes and antenna are absent. Okay, what is the type of metamorphosis you see in this kind of insects is that they don't have, they increase their body number, or the appendages or the abdominal numbers are increased during each molt. Because of that, we call them as anamorphosis. Clear? Is it clear?
Yes, sir.
Okay.
Yes, sir.
See, I know it is not, you need some time to get into this. Here afterwards, we will be studying all the insect orders, 30 insect orders. And for all the 30 insect orders, you should be able to remember the characters. How do you write? They'll ask you, write the write 2 or 3 important characters of the order Protura. So what will you write? So means you should be able to write the antenna and eyes are absent. Okay. Abdomen 11 segmented, anamorphosis, and 4 legs are sensory, are held above the head. So if you write 4 characters, you can easily get marks. So eyes and antenna are absent. You can easily remember that. So that is how you can remember the character. Protura means, pro means primitive. Okay, so protuberance. Okay, no, pro means in the front, right? So you can see that the legs are kept in the front. So because of that, we call them as protuberance. You can see that in this diagram also, in this photographs also, the antenna are absent. Eyes, there is no eyes. Okay, so no antenna and eyes are absent, but still the legs are kept like in the front. Okay, and they have a terminal telson in the last few abdominal segments. Okay, and there is no growth. Every time they mount, the number of abdominal segments will be adding. Anamorphosis, define anamorphosis. That is the character, or that is the define anamorphosis. Every mount they add the abdominal segment. Only the abdomen segments will be added during every mount. That is what we call it as anamorphosis. Clear? Okay. So there is, that's how they look in live. If you put, if you look at in the, they are present in the organic matter. You can see that there is no eyes in this also. Four legs are held like antenna. Eyes and antenna are absent. Okay. Protura, eyes and antenna absent. Finished. That's how you identify that. Okay. Then coming to the second order, that is diplura. Di means two. Plura means what? Okay, I'll tell what it is. Antenna are moniliform. Where do you get moniliform antenna?
Termite.
Very good.
Termites.
Yeah, good, very good. So you can see that the moniliform antenna with, okay, compound eyes and ocelli are absent. There are no eyes. Eyes are absent in case of the diplurans. Okay, protura, eyes and antenna both are absent. Whereas in case of diplura, only eyes are absent. Okay. And what is important is abdomen is 10 segmented. Whereas in case of the protura, the abdomen is 11 segmented. And in case of this 10th segment, the last segments are with a pair of styli. Okay. There is a pair of styli, and also they have a pair of irreversible vesica on at least some segments. Okay, I'll tell you what these irreversible glands are. Okay, larval development is epimorphous. Epimorphic means the larva will also have same number of segments. If even you take first instar, second instar, or adult, the same epimorphic development is seen. Okay. Example is in India Japix indicus. It is from India, so they have named it as indicus from India. A species of diplura from India is India Japix indicus. Don't worry about the examples right now, okay? But only look for the characters. Protura, eyes and antenna absent, whereas abdomen is 11 segmented. Whereas diplura, in case of diplura, only eyes are absent and the abdomen is 10 segmented. Clear? Antenna is moniliform. So that is how you can differentiate. Then coming to these are the irreversible glands. Okay, irreversible vesica, which is there on the ventral surface of the abdomen. Don't worry about that. Okay. So coming to the last one. So that is the third insect order, non-insect order. That is the columbola. So what is columbola? You know, right? Columbolans are called as springtails or snow fleas because they jump. Antenna usually 4 segmented, or the first 3 provided with okay, they are usually 3 or 4 segmented antenna. Usually they are 4 segmented. Compound eyes absent. The compound eyes are absent. Only the compound eyes are absent. Abdomen with 6 segments. So in case of protura, the abdomen is 11 segmented. In case of diplura, it is 10 segmented. In case of in case of the columbola, the abdomen is 6 segmented. So that means very interesting. So whereas the 3 segmented 6 segmented abdomen is again modified. They have pregenital appendages. So they have colophore on the first abdominal segment. You can see that there is a colophore there. Okay. Then minute retinaculum on the third. Okay. 1, 2, and 3. There is a retinaculum here. on the third, and four springing organ on the fourth abdominal segment. So this is the fourth abdominal segments. They have a forking organ or a springing organ, or it is like a fork. Okay, the furcula helps the columbolans to leap in the air. Sorry, I'll 1 more minute, I'll finish it off. So this is how the columbolan looks. Okay, very, very tiny fellows. You can see this furcula, colophore. Okay, so this is how in life they look. Okay, very, very tiny creatures. They look very cute, but you need to have a hand length of 10x them under the in the soil. Okay, so you can see the colophore there. Sorry, this is colophore. This is the retinaculum, forked. There are 2 fork. Okay, so this is how they look. Very, very interesting tiny creatures. Can you see the colophore there? Okay, retinaculum. Okay, I think you should I think I'll stop at this. Thank you.
Thank you, sir.
Yeah. I'm sorry, it's too much for an holiday.
No problem, sir. Thank you, sir.
Yeah.